Hello, my name is Caitlyn Thomas, and the title of my project is Using a Molecular Modeling and Native State Mass Spectrometry Quick Screening Approach to Understand the Inhibition of IMP1 Variants. You can see the list of co-authors there, and I work for Dr. Michael Crowder in the Department of Chemistry and Biochemistry. I'll begin with a brief introduction. Bacterial infections are commonly treated using beta-lactam antibiotics. However, due to overuse, antibiotic-resistant bacteria have evolved. Approximately 2 million people are infected with antibiotic-resistant bacteria, and 23,000 people die each year due to these infections. One way bacteria become resistant to beta-lactam antibiotics is through the production of beta-lactamases. Beta-lactamases are capable of hydrolyzing the beta-lactam bond, which renders the beta-lactam antibiotic inactive. This study will focus on a subgroup of the beta-lactamases called metallobeta-lactamases, or MBLs for short. In order to combat MBLs, scientists are trying to create an inhibitor which can be paired with a beta-lactam antibiotic. However, no inhibitors of MBLs have been created, which makes this study a very important topic. My study will focus on the MBL IMP1 and the variants of IMP1. I have created a quick screening approach to efficiently study whether an inhibitor of IMP1 is also capable of inhibiting the variants of IMP1. In order to achieve this, I will use molecular modeling and native state mass spectrometry. The first results I will present are from the molecular modeling study. I have chosen to focus on IMP78 because it has a double mutation close to the active site, which could prevent an inhibitor from binding. In figure 1A, you can see a pi-mole molecular visualization model of IMP1 with an inhibitor bound. The inhibitor is binding to or near the 2 zinc ions in the active site of IMP1. In figure 1B, you can see a pi-mole model of IMP78 with the same inhibitor bound. There are also many red circles shown in the figures. These circles represent any clashes or bad interactions between the protein and inhibitor. You can see that the clashes are very minor with IMP1, but may be a little more severe with IMP78. In order to better determine the severity of the clashes, I used a software called Molprobity to quantify the clashes. The results are shown as a heat map in figure 2. You can see that the color of the clash score for both IMP1 and IMP78 for inhibitor 3R9D are fairly similar in color. This means that the inhibitor may inhibit IMP78 like it does IMP1. In order to determine if the inhibitor does inhibit IMP78, native state mass spectrometry was used. In figures 3 and 4, you can see the mass spectra for IMP1 and IMP78 respectively. The largest peak in both spectra corresponds to the mass of IMP1 or IMP78 with 2 zinc ions bound. Because this is an ongoing study, there are multiple experiments remaining. The next step will be to use native state mass spectrometry to analyze each protein and inhibitor combination. Then isothermal titration calorimetry will be used to determine the binding affinity each protein has for the inhibitor. Lastly, an IC50 will be determined for each protein and inhibitor combination. In conclusion, this study will help to guide the design of future inhibitors of MBLs in the hopes of designing a pan inhibitor of MBLs.